Let me begin today's lecture with the following simple proposition. How living systems use energy to create and maintain order can largely be understood as a matter of chemistry. Much of this chemistry is a matter of building polymers of biomolecules, those biomolecules we introduced early on in the course, including carbohydrates, those are sugars, nucleic acids such as DNA, proteins, and lipids. By this I mean making larger molecules out of smaller ones. For example, proteins are polymers made up of amino acids. We'd call the amino acids the monomers. Similarly, nucleic acids such as DNA are polymers made by joining together nucleotides. The energy needed to build these polymers is energy that's invested in the creation of a greater degree of orderliness on the molecular level. But living systems don't just build things, they also take them apart. This process can be seen as sort of the breaking down of polymers back into their building blocks. It also involves the breaking down of all sorts of complex biomolecules into smaller, less complex parts, in fact, sometimes down to their basic elements. You can think of the cell as a kind of miniature chemical industry. Some chemical processes are involved in breaking down resources that come into the chemical plant, that come into the cell, that is, breaking them down into their constituent components, their chemical building blocks, which could be used for other things. Other chemical processes are involved in building new compounds out of these raw materials, these building blocks, making compounds that are going to be used by the cell in other ways. The term metabolism refers to the sum total of all of the biochemical processes that occur in an organism's cell. In a typical cell, there are literally thousands and thousands of chemical reactions that are able to occur, all interrelated through intricate pathways that connect them. Now, if you looked at a schematic diagram of even just a few hundred of the most important of these thousands of reactions, you tried to sort of map out how these reactions were all related to each other, you'd see something that might look like the diagram of an incredibly complicated subway system. Hundreds of chemical compounds would be connected to other compounds by the particular biochemical reactions that transform one compound into another. Just like dots representing the stations on a subway map are connected to each other by different rail lines. Some compounds may only be connected to the compounds that immediately precede them or follow them in a fairly lengthy pathway. While other compounds, in fact many compounds, would be connected to at least several other compounds, maybe even dozens of other compounds with branching and intersecting pathways connecting them. Now, if we added up all of the interconnected pathways, biochemical pathways found in this cell, not just the key pathways, and put all of these on some sort of map of their interrelationship, we'd actually see something that looked more like a tangled bowl of spaghetti even than a complicated subway map. Now, the point of this description of a schematic diagram of the metabolic pathways found in the cells is to impress on you not simply the fact that the biochemical processes found in living cells are pretty complex, but more importantly, that all of these biochemical processes are potentially interrelated. They connect to each other. The product of each reaction is likely to be the substrate of another reaction. And all of these products and substrates are may themselves be important end products that perform some other critical function that the cell needs to have done. The biochemistry of cellular metabolism is like a beautifully choreographed ballet. It's complicated to look at schematically, but it all works together in a very, very neat and organized fashion, connected through these interconnected chemical reactions. Now, we only have time to take a look at a few parts of this ballet of metabolism in this course. And so, over the next few lectures, what we're going to do is focus our attention on those metabolic processes that are particularly important for processing energy in the cell. So, let's begin our look at the essential chemistry of life. Begin our look at how metabolism works by considering the basic energy relationships of biochemical processes in general. Metabolic reactions can be subdivided according to whether they break down complex compounds into simpler ones or alternatively build simpler compounds into more complex ones. We refer to those metabolic processes that break compounds down into smaller components as catabolic pathways. In general, catabolic pathways release energy. That is, when a catabolic reaction runs, some energy is produced and released. We say that these catabolic reactions, therefore, are exergonic. Exergonic is a term that means literally energy outward, and thus it refers to reactions that give off energy when they run. Now, the most important set of catabolic pathways, exergonic pathways, that we need to understand are those that are involved in the breakdown of sugars, which is what makes the energy that will be used to produce ATP, which is where we're going with this in the long run. This set of metabolic pathways, the ones that break down sugars and some other organic compounds, are known as cellular respiration. And we're going to begin discussing those pathways in detail in a couple of lectures. Now, we refer to the metabolic pathways that build complex compounds out of simpler building blocks as anabolic pathways. In general, anabolic pathways require energy. That is, an anabolic reaction needs some energy to be put into it in order for that reaction to run. We say that these anabolic reactions, therefore, are endergonic. Endergonic means energy inward. So endergonic reactions require that energy be put into them if they're going to run. Anabolic pathways include those pathways that we've discussed quite a bit now that are involved in the synthesis of proteins, for example, out of amino acids, or the synthesis of nucleic acids from nucleotides, those things that we've discussed earlier in the course. The key point to keep in mind here is simply that catabolic reactions release energy and anabolic reactions require energy for them to run. Therefore, much of what we want to understand about energy in living systems on the molecular level is how the energy that's released by catabolic pathways can be harnessed and stored and transferred for use to run anabolic pathways. Before we go any further, I want to give you a better idea of what I mean by energy in this context. Now, strictly speaking, energy refers to the capacity to do work. That is, energy is the capacity to move matter against some opposing force. For example, it takes energy to lift an object. In this case, the opposing force is gravity, and energy is needed to do the work of lifting that object up against the force of gravity. We can also think of energy as the ability to rearrange a collection of matter that's resistant to being pushed around. In terms of chemistry, and these are the terms that are really most relevant for our discussion right now, we can think of energy as the ability to rearrange molecules by building or breaking chemical bonds, the chemical bonds that hold those molecules together. Those bonds are resistant to being broken, and those molecules, therefore, are resistant to being rearranged. We need energy in order to do that. Energy comes in many forms, of course. There's chemical energy, there's mechanical energy, there's electromagnetic energy, and we don't really need to concern ourselves too much with these details here. There are a couple of points I want to make, however. First, I want to remind you that all forms of energy can be considered to be one of two basic types. Kinetic energy is the energy associated with movement or activity. It's kinetic energy that actually does the work that does work by, for example, altering the motion of some matter or changing the properties of some matter. Potential energy is stored energy. Potential energy can be stored in many ways, such as in the position of a piece of matter resisting a force. Or, as we'll see to be especially important in biology, potential energy can be stored in chemical bonds. And it also can be stored in terms of a chemical concentration gradient across a membrane. Now, a second important point I want to make here is that not all the energy in a system is really available to do work. I don't want to go into the physical details here, but I want to point out that we refer to the energy in a system that can be used to do work as the free energy of that system. Now, for the sake of simplicity, later on in our discussion, I might sometimes refer to free energy just as energy. I might say the energy of this compound is such and such relative to that. What I really mean here, and I want to remind you, is the free energy of that compound or that biological system. And that free energy is the energy available in that compound or that system that can actually do some sort of work. To a physicist, that might be too vague a definition, but I think for our purposes here, it'll be fine. Now, here's the important take-home message. The amount of free energy a system has, or what we'll just call energy occasionally for simplicity's sake, the amount of energy that a system has is related to how stable or unstable that system is. Or to put it another way, it's related to how likely that system is to change its state spontaneously. Systems with a lot of free energy are relatively unstable, and thus they're more likely to spontaneously change in some way. Systems with relatively little free energy are more stable, and thus they're unlikely to change in any way spontaneously. Now, we also tend to think of high-energy systems as being more orderly. That is, a high-energy system is in a more orderly state, whereas a low-energy system is in a more disordered state. This corresponds to our intuitive understanding of the relationship between energy and order in biological systems that we introduced last time. Energy is required to establish and maintain the orderly state of living things, and if they don't have energy, they become disordered. Okay, let me give you some examples to make clear the relationships between free energy, orderliness, and stability. Now, one relatively intuitive example we might think about is the force of gravity acting on an object. Let's say we have a ball on a ramp, or maybe a child on a playground slide. A ball at the top of the ramp is in a high energy state relative to a ball at the bottom of the ramp. Similarly, we might say that a child who's at the top of a slide is in a higher energy state, physically speaking, than a child who's gone to the bottom of the slide. Now, how do we intuitively know this is true? How do we intuitively know that there's a difference in energy level? Well, the reason is that if the ball is at the top of the slide, it will spontaneously roll down to the bottom. It goes from the high energy state to the low energy state. But the reverse won't happen. If a ball is at the bottom of the slide, it won't spontaneously roll back up. Now, if the child's at the bottom of the slide, it might spontaneously, behaviorally speaking, run back up. But we know it's using energy to do that, right? It's using the chemical energy that is making its muscles work. Now, this example illustrates how the high energy state of a system is inherently less stable. If we have a ball at the top of the ramp, it will spontaneously roll to the bottom, but not vice versa. A less intuitive example, but one that we've already encountered in our course, comes from thinking about concentration differences across a barrier or membrane, the concentration difference of some substance. We've seen this in a couple of contexts. For example, when we were talking about how neurons work. An ion that's more concentrated on one side of a cell membrane will tend to spontaneously move down its concentration gradient to the other side of that membrane. This, of course, assumes that the membrane is what we call semi-permeable. That means that the ion could actually cross the membrane. In this case, having a large concentration difference between the two sides of the membrane represents a high energy state. Having the same concentration on both sides of the membrane represents a low energy state. Again, this difference in energy state is apparent from the fact that molecules will spontaneously move from the high concentration side to the low concentration side, but not vice versa. And remember, when we were talking about neurobiology, we pointed out that there are some proteins that act as active ion channels that will pump those ions up their concentration gradient, but they need energy to do so. In fact, they get that energy from ATP. Now, a final example to consider is to go back to thinking about the structure of molecules. A more complex molecule generally represents a higher energy state and therefore a less stable state as compared to simpler molecules built from the same atoms. This example is even less intuitive, but it sets up an important point. For example, consider a molecule of sugar. Let's take a kind of sugar called glucose to be specific. As we'll see in more detail later, this glucose molecule is made up of a bunch of carbon atoms, hydrogen atoms, and oxygen atoms arranged in a particular way. Now, the exact same number of carbons and hydrogen and oxygens can also be rearranged to form several molecules of carbon dioxide and water. Even though glucose on the one hand and some molecules of carbon dioxide and water on the other hand are made of exactly the same stuff, the same number and kinds of atoms, glucose, being more complex, is more orderly and has more energy stored in it, whereas the carbon dioxide and water, being less complex, have less energy, and we refer to them as being less orderly, from a molecular point of view at least. Consistent with our earlier examples, the higher energy sugar molecule is more likely to spontaneously break down to form carbon dioxide and water than vice versa. In other words, you will not get carbon dioxide and water spontaneously rearranging themselves to form glucose, but glucose might spontaneously break down and rearrange itself to form carbon dioxide and water. Now, just a little aside here. Actually, glucose doesn't spontaneously do this easily, but that's a different problem that we're going to talk about later in the course. It might do this, and it's certainly more likely to do this than vice versa. Glucose is high energy. It can be broken down more easily into carbon dioxide and water than vice versa. Okay, now let's get back to thinking about energy relationships in metabolism. As I told you, catabolic reactions generally break down more complex compounds into simpler compounds. In this sense, these catabolic reactions transform compounds that are in a high energy state into compounds that have a lower energy state. Let's consider what this would look like if we drew a graph of the change in energy that occurs as a typical catabolic reaction proceeds. Let's let the x-axis of this graph represent time, which is just the course of the reaction as it runs from start to finish. And let's let the y-axis of this graph represent amount of energy, amount of free energy, running from a low energy state on the bottom to a high energy state at the top. Now let's place two symbols on this graph. One symbol is going to represent the molecule or compound we begin the reaction with. We're going to call this the reactant of the reaction. The other symbol represents the compound that we're going to end up with after the biochemical reaction has run. We're going to call this the product of the reaction. For a catabolic reaction, we would place the reactant on our graph in the upper left at the high energy end, and we would place the product down in the lower right, the low energy part of the scale. In other words, our graph of energy relationships of a catabolic reaction would have a line going from high energy to low energy. In other words, catabolic reactions energetically run downhill, if we think of it graphically. Actually, if we think of this graphically, this is like thinking about a ball rolling down a hill. Thus, we expect these catabolic reactions to run spontaneously. And furthermore, because they go from a high energy state to a low energy state, we also expect them to release energy when they do occur. Now, by contrast, anabolic reactions transform compounds that are in a low energy state into more complex compounds that have a higher energy state. That is, they transform a simple compound to a more complex compound at a higher energy level. Now, if we graph an anabolic reaction on the same set of axes, then we would start with the reactant down in the lower left-hand corner, down in the low energy end of things, and we would end up with the product of the reaction in the upper right corner. In other words, anabolic reactions run uphill from an energy point of view. We start with a low energy reactant, and we end up with a higher energy product. Graphically, this would be like our ball rolling uphill. Thus, we expect that anabolic reactions will not occur spontaneously, just like the ball won't roll up the hill spontaneously. And furthermore, we expect them to require energy to make them run. We have to add energy to get that product at a higher energy state. Now, the reason I've spent some time emphasizing the difference between catabolic reactions, which run downhill energetically, and anabolic reactions, which run uphill, is because these relationships help us to define a central question we need to address when we're talking about energy in living systems on the cellular and molecular level. And the question is simply this: how do we get anabolic reactions to transform reactants from a low energy state into products at a high energy state? This is a tough problem for the same reason that a ball doesn't naturally just roll up a ramp spontaneously. Now, over time, in general, systems tend to go from states of high energy to states of low energy spontaneously, because they will, in general, roll downhill spontaneously. But the reverse doesn't happen. Now, another way you could state that is to refer to what's called the second law of thermodynamics in physics, which predicts that disorder in any system will increase over time. Disorder, a lower energy state, is going to be the likely outcome of any system. The energy will run down. Intuitively, then, we see that there's no big problem to understand, at least to a first approximation, how catabolic reactions might run in a cell, because they will run spontaneously downhill from an energetic point of view. Although we'll see later that there's another problem lurking here that we have to deal with these catabolic reactions. Catabolic reactions, therefore, are consistent with the second law of thermodynamics. They take a system from a more orderly, higher energy state to a less orderly, lower energy one. But how do we get anabolic reactions to occur at all? How can we make such reactions to run uphill? At first glance, to restate this problem, anabolic reactions appear to defy the second law of thermodynamics. They take systems from low energy states and put them into higher energy states. And remember, these anabolic reactions are really essential for life to occur. These are the reactions that are building the stuff that we made of, things like proteins, things like DNA, and so forth. Clearly, cells have to get anabolic reactions to run, otherwise life couldn't exist. In fact, one might characterize life as being as something that defies the second law of thermodynamics. If you look at physical systems in general, they will generally go to more disordered states over time. But living systems generally go to higher ordered states and maintain this order in apparent defiance of the second law of thermodynamics. Of course, we know that living things can't really defy the laws of physics. Instead, their trick is a relatively simple one, and you might have guessed it already. In order to create and maintain an orderly state, what living things do is simply take some energy and add it into the system. They get energy from someplace else and pump it in. Using this energy that they add to the system, living things can change that system from a less orderly, lower energy state in general to a more orderly, higher energy state, and maintain that orderly state over time. So, to get an anabolic reaction to run uphill, all we have to do is add energy somehow. This is the same as if we want the ball to roll up a hill. All we have to do is add energy, in this case the physical energy, to do the work to roll it up the hill. But then this leads to the two questions we now need to start addressing. The first question is, where does this energy come from? The second question is, once we've got this energy, how do cells actually add the energy in to make these anabolic reaction runs? How do they essentially push these reactions uphill from a biochemical or a molecular point of view? Now, the answer to the first question probably has already occurred to you. Remember, catabolic reactions release energy. In theory, then, at least, you might imagine that what a cell can do is obtain energy by running catabolic reactions, those energy-releasing exergonic reactions, and use this energy to cause those anabolic reactions to run uphill. So the source of energy might be from catabolic reactions. This sounds simple enough, and in fact, cells do something like this. Although it turns out, as we'll see shortly, that capturing the energy from catabolic reactions can be a little tricky. We're going to skip over that problem from now and return to it in some detail in a couple of lectures. Let's assume for the moment that the cell has obtained energy from catabolic reactions and can use this energy. Now what we want to do is to turn to the second question. How specifically is this energy used to drive an anabolic reaction uphill? Here's where the molecule ATP comes into play. The key strategy used by living systems, by cells, to drive anabolic reactions uphill is through a process we'll refer to as energy coupling. The energy that's released by catabolic, exergonic, that's energy-releasing reactions, is stored in a molecule, the molecule ATP. And then that molecule of ATP becomes involved in the anabolic reaction itself. It becomes really one of the substrates of that reaction. And the energy of ATP that's stored in ATP is used to drive that anabolic reaction uphill. Now, to begin to understand how this works, let's consider the molecule ATP in a little bit more detail. ATP is related to the nucleotide adenine. Remember, adenine is one of the nucleotides, one of the 4 nucleotides we saw when we were talking about nucleic acids such as DNA and RNA. But it differs from adenines that we saw when we were talking about nucleic acids by having 3 phosphate groups attached to the ribose sugar instead of just 1, which is why we call it adenine triphosphate. Remember, a phosphate group is just a phosphorus atom bound to 4 oxygen atoms. Now, this detail in itself isn't important, but what is important to know is that the bonds between adjacent phosphate groups in this triphosphate tail of ATP store a lot of energy. They're very high energy bonds. Furthermore, the high energy bonds, these high energy bonds between phosphate groups are not completely stable. The chemical bonds between the terminal phosphates, these chemical bonds between the terminal phosphates of ATP can be broken, yielding a molecule of ADP, that's adenosine diphosphate, just 2 phosphates, and a separate inorganic, we'd say, phosphate group. In other words, one of the 3 phosphate groups can be cleaved off. The biochemical process that does this in general is referred to as hydrolysis because, as it turns out, the breaking of this bond, which is where the term lysis comes in, requires a water molecule, which is where the hydro comes in. So hydrolysis is the breaking of this bond. Now, the most important point to keep in mind is this. The hydrolysis of ATP into a molecule of ADP and an inorganic phosphate group is a very, very highly exergonic reaction. It releases a lot of energy. And it's the ability of ATP to store and release this energy by adding and subtracting phosphate groups that makes ATP a particularly useful energy currency in the cell. You can kind of think of the triphosphate tail of ATP as being sort of the chemical equivalent of a loaded spring. With the 3rd phosphate added, this spring is compressed, storing energy, and when you cleave this phosphate group off, it's like releasing the energy in the spring. Because energy can be stored in ATP, living systems can couple this energy, which was obtained from catabolic reactions, with anabolic reactions to make them run uphill. Now, let's just discuss a little bit how ATP actually does this. There are actually a number of different ways that ATP might donate its energy to a reaction or to some cellular process, but the most fundamental one involves actually attaching one of the phosphate groups to one of the substrates of the anabolic reaction being run. And in this sense, what ATP does is it donates its energy in a 2-step process. Consider, for example, a chemical reaction that converts glutamic acid, which is a kind of amino acid, into glutamine. And it does this by the addition of an ammonia molecule. Now, I won't detail you or bother you with the specific details of the chemical structures here, since it really doesn't matter. But you can essentially think of this as a chemical reaction that starts with 2 compounds and then joins these together to form a single compound, the compound glutamine. This reaction is anabolic. It requires energy to run. And so it wouldn't normally run spontaneously. But cells have a way of making this reaction run uphill by separating it into 2 steps as follows. In the first step, a phosphate group is transferred from an ATP molecule to one of the substrates, the glutamic acid molecule. This step is exergonic because the hydrolysis of the phosphate from ATP releases so much energy. And so this step will run energetically downhill. The energy that is released from this process will actually attach that phosphate group to the glutamic acid. That phosphate group now carries with it some of the energy that was stored in it. So the glutamic acid itself now is at a higher energy state. We refer to this as being phosphorylated. And this phosphorylated intermediate, as we would call it, is a much higher state than the original substrate that before the phosphate group was added. Now, as a result, we're now got a reaction that would combine a phosphorylated glutamic acid, which is a very high energy molecule, with a molecule of ammonia. This reaction is actually an exergonic reaction. It now will run energetically downhill. The reason it does so is one of the substrates of this reaction, that phosphorylated glutamic acid, has so much free energy in it because of the added phosphate bond that the combination of that now phosphorylated substrate with the other substrate, the ammonia, has got a different energy relationship. The energy in the phosphate bond will now be released when these 2 molecules come together, and that release of energy will drive what was an endergonic reaction uphill. it's effectively changed an endergonic reaction into what's really an exergonic reaction. Well, although this coupling of energy derived from hydrolyzing ATP can drive endergonic reactions uphill energetically, it's important to remember that we're not really violating the second law of thermodynamics in any real sense. What organisms are doing to drive these anabolic reactions, to create greater order, to make systems have higher energy levels, is to take energy from someplace else and add that energy into the system. The currency of that energy is often ATP. But once we've used ATP, we've used up the energy. We now have a molecule of ADP, adenosine diphosphate, which is a much lower energy molecule. Fortunately, we can make more ATP out of ADP. That is, ATP is a renewable resource. But to renew ATP, we need yet another source of energy. And we have to ask where that energy comes from. Before we do that, though, in the next lecture, what we're going to do is turn our attention back to enzymes and ask how enzymes make these kinds of reactions run in general.